So we have talked about some of machinery which is responsible for moving proteins across the nuclear membrane. We have talked about the characteristic of the nuclear pore. We have talked about the sequence on the proteins which targets them to the nucleus. Now let's see the rest of the machinery which captures these proteins that have NLS or nuclear localization sequence and then brings them or imports them into the nucleus. And we'll see how energy is used to bring these proteins against their gradient, because we are actually going to decrease entropy. So how does that work? Let's look at that. Nuclear import receptors, these are the proteins which can carry proteins which have NLS. There are several different types of these nuclear receptors. In order to initiate nuclear import, NLS has to be or must be recognized by a nuclear import receptor, and these receptors are encoded by a family of related genes. And actually we will also talk about export receptors. They're also similar and they also come from the related genes. Each family member encodes a receptor, a protein that is specialized for transport of group of nuclear proteins sharing structurally similar nuclear localization signals. So we have various different types of nuclear import receptors that basically are specialized in recognizing a specific type of NLS. After these receptors have bound their cargo or proteins that have NLS, they basically bind the nuclear pore. Nuclear pore inside the channel basically is lined with FG repeats. FG repeats are basically phenylalanine and glycine. We know these are two amino acids. The nucleoporins, which are making the inner surface of the nuclear pore, have abundant phenylalanine and glycine residues. So these nuclear import receptors, once they have bound their cargo, they bind these FG repeats which are present inside, which are lining the nuclear pore, and they basically interact with them and they sort of crawl on these FG repeats. So these proteins, our nuclear receptors, they repeatedly bind and dissociate from these FG repeats, thereby crawling inside the nucleus. Once our nuclear receptor and its cargo have reached the nucleus, they basically dissociate, the cargo dissociates, and the nuclear import receptor is recycled by sending it back into the cytoplasm. Sometimes the nuclear receptors don't bind their cargo directly. They can use also adapters. This basically gives the cellular machinery or the system more versatility, so they can bind different types of proteins, different types of cargoes, and they can transport them inside the nucleus. So this is basically, use of adapters basically increases the versatility of the import machinery. Nuclear export receptors also work like nuclear import receptors, but in reverse, because they're going to bind their cargo and they're going to release in the cytosol, which is basically reverse of what nuclear import receptors are doing. Nuclear export receptors bind large molecules. For example, we have talked about ribosomal subunits and RNA, which can also be a large molecule, through the nuclear pore, and it is also a very selective process. Nuclear export signals are present on molecules to be exported and by the use of nuclear export receptors. These receptors bind both the export signal and the nucleoporins. We saw that our import receptors also bound the cargo and the nucleoporins, and specifically the FG repeats on the nucleoporins. And they guide their cargo through the pore complex and ultimately taking it to the cytosol, where the cargo is released. So export receptors and import receptors are structurally related and encoded by the same family of genes, which are also called karyopherins. So there's no free lunch, as we know. So we have to spend energy if we are moving molecules against their gradient. So where does the energy factor come in? I have to introduce you to a molecule. It is called RAN. This RAN molecule is also a G protein, meaning that it binds GTP and or GDP. So like our other friend, our G protein, it can be bound to GDP or GTP, meaning it sort of works like a switch in an on position or in off position. When it is bound to GTP, our RAN is in on position. When it is bound to GDP, it is off position. So this basically also, when RAN hydrolyzes GTP like our G protein does, we have talked about, for example, RAS or G proteins, when they hydrolyze GTP, they basically use up energy. So this is where the energy is coming from, and this is what basically drives the import and export process. Now, RAN is found both in the cytosol and the nucleus, and it is required for both import and export from the nucleus or into the nucleus, but there is a trick to it. We'll see what the trick is. It basically, as we know in RAS, there are molecules, accessory molecules which help RAS, and actually there are also two molecules which help RAN also. So we have talked about gaps and JEFs. So there's a gap, RAN gap, which is basically RAN GTPase activating protein, and RAN JEF, which is RAN GTP exchange factor. So basically the gap is present in the cytosol, and strictly RAN JEF is present inside the nucleus. So basically in the cytoplasm we will have the RAN GDP, RAN bound to GDP, and inside the nucleus we will have RAN bound to GTP. So because the RAN JEF is here, and it is going to allow RAN to exchange GDP for a GTP molecule, and RAN gap is going to activate the enzymatic activity of RAN, and it is going to allow RAN to hydrolyze GTP into GDP, and of course a phosphate ion will be released. Okay, so how does this process basically relate to our nuclear import? Let's look at that. Here, for example, we have the receptor, nuclear import receptor. It will bind the cargo protein, and when it does so, it goes through conformational change which allows it to enter the nucleus. When it enters the nucleus, here it will bind RAN GTP, which will allow this receptor to unload its cargo, and RAN GTP bound to the import receptor allows it to again bind FG repeats and actually go back to the nucleus. So we will talk about this process in more detail, how the import and export processes are facilitated by RAN, and how the export receptor and the import receptor are able to selectively bind proteins and release them in appropriate compartment.